So in this lecture we'll be talking about blood biochemistry. And these are the following learning objectives. Firstly, we'll be talking about what's the role of blood in the body, what's the characteristic of blood, and what are the cellular as well as the plasma components of blood, how is blood a vehicle for transportation in the body, and what are the hemostatic functions of blood, and how is heme formed and degraded, and we're going to also talk a little bit about blood typing. Firstly, when we talk about blood, blood is basically a connective tissue, and as with all connective tissue, it has cells and the extracellular matrix. Here, the cells that we can find in blood consist of the blood cells that we know as red, white blood cells, as well as platelets, and the extracellular matrix is basically the blood plasma. And blood has so many different functions in the body. Firstly, for protective purposes, for transportation, and for regulation. Exactly how does it protect? Well, there's firstly a protection against losing blood, which is called hemostasis, and then there's also protection from pathogens, and this is what basically our immune system does. But as I've said before, that's because blood is composed of white blood cells. These white blood cells have a big function in the immune system. And then for transportation purposes, blood can carry oxygen, hormones, as well as nutrients. And for regulation purposes, blood is an important mediator for homeostasis of acid and bases, and also to maintain electrolyte balance and body temperature. Now, in the human body, we can find about 4 to 6 liters of blood depending on the weight and the age of the person. When we look at the color, usually venous blood is darker than arterial blood. The constant pH of blood is between 7.35 to 7.45, so it's a very narrow and constant range. And then the viscosity of blood is usually 3 to 5 times more than water. And then when we look at the components of blood, as I've stated before, there's basically 2 main components of blood. We have the plasma and we have the cells. You can see it when we centrifuge a blood down that the plasma will usually accumulate at the top, and then the cells will just accumulate at the bottom along with the other cells. And here the plasma component makes up about 50 to 60% of blood, while the cellular components make up the rest. And when we talk about the plasma component, it is majorly composed of water. So water makes up about 95% of the plasma, and the remaining 5 to 9% in plasma is basically proteins, nutrients, electrolytes, waste, and hormones. So what kind of proteins can we find in plasma? Well, basically just carrier proteins mostly. These are the proteins that carry a lot of other molecules, whether it's fatty acids, cholesterols, bilirubin, copper, etc. There's also other proteins that are involved in blood clotting, and there's also globulins, which are part of the immune system. And then when we talk about nutrients, of course, we've got the amino acids, sugars, and fatty acids that are obtained from our diet and are spread around throughout the body for a lot of metabolic purposes. And then for electrolytes, we also have them in plasma, and these are maintained at a balanced state in order for our pH to be stable, as well as our osmotic pressure to be stable. And then we have the waste products in blood, usually not really urea, but mostly, not only urea, but also creatine and lactic acid and carbon dioxide. And finally, we also have hormones, which regulate a lot of functions in the cells and organs. So these can also circulate and travel in blood in order to perform the endocrine functions. This is just a little reference for everybody. So as you can tell, because plasma carries a lot of things and a lot of metabolites, there's actually a saying that plasma is the window into metabolism, because a lot of the biochemical tests that we do in the hospitals are done on plasma, the plasma of the blood. And to do that, usually we take the blood samples into the lab. They're taken in a special tube which doesn't allow the plasma to clot, and then we centrifuge it down, as you can see from the previous picture, so that we can separate the plasma from the blood cells. And then we can measure so many different things in the plasma. We can measure the metabolites such as glucose, lactate, pyruvate. We can measure the waste products like urea, creatinine, a lot of other things, and also the lipids. So this can tell us a lot about the metabolic state of a person based on the concentrations of metabolites in plasma. Now, when we talk about blood components, there's also, of course, cellular components. And these can be divided into three main types of cells. There's the red blood cell, the white blood cell, and then there's the platelet or thrombocyte. And this is just a little reference for everybody as well. What are the normal ranges for the blood cells in our body? And as you can see, all of our blood cells, even though we have a lot of different types, they basically originate from the same stem cell in our bone marrow, and this is called the hematopoietic stem cell. And the process of making a blood cell is called hematopoiesis. And this is just a little term that we need to be familiar with, because hematopoiesis is then regulated by several different mediators and several different factors. But in fact, the process itself is a general process which all stems from the hematopoietic stem cell and to generate blood in the process called hematopoiesis. So let's talk about the cells one by one. We probably won't talk too much about the white blood cells because we'll have a special lecture on immunology just for that. But we'll focus on erythrocytes and their function, as well as thrombocytes and their function. So let's start with erythrocytes. This is a picture of an erythrocyte, or a red blood cell. And this is the predominant portion of blood cells. A major percentage of the blood cells in our body is actually erythrocyte. And the main function of erythrocyte, as you may know already, is to transport oxygen and to remove carbon dioxide and hydrogen ions. And the reason why they're capable of doing this is because they've got a special protein called hemoglobin inside. So when you look at an erythrocyte, each part of the erythrocyte is made of a lot of hemoglobins, and each hemoglobin has a heme group, which has an iron inside that is bound to an oxygen. So that's the reason why the major function of the erythrocyte is to transport oxygen. And to do this, it's got a special shape. Firstly, the shape, it's got a lot of special characteristics. The first is the shape. The shape itself is biconcave. The reason for its biconcave shape is that it can increase the surface area and the efficiency of oxygen exchange, and it allows the cells to better squeeze through the tiny little capillaries. And then if you look at an erythrocyte, there are no organelles in there, so there's a lot of free space for the hemoglobin, and it does not require cellular respiration, meaning that it will not require any energy. So no additional energy for the organelles, because there is no organelle. And then it's composed of the protein hemoglobin, in which one hemoglobin can carry 4 oxygen molecules. And there are approximately 250 million hemoglobins in every red blood cell. And the production of red blood cell is regulated by the hormone erythropoietin, and it has a lifespan of 120 days. Now, the second cell component of blood is leukocyte, or white blood cell, and this only makes up about 1% of the cells in blood, but this percentage can actually increase when there's an infection. And the main function of leukocyte is as an immune cell and to fight against a lot of different pathogens. And as I said before, we probably won't go too much details into these leukocytes, because we have a separate lecture for these, which is the biochemistry of immunology. So I'll move on to the last blood cell, which is platelets or thrombocytes. So if you look at a platelet, they are also circular cells, and also they don't have any nucleus, just like the red blood cell. So they are anuclear. And the diameter is very small, about 2 to 3 microns. And the interesting thing about platelets is that they can actually change their shape. So this is a platelet that is not active, and this is an active platelet. So when it needs to get to work, it changes its shape into this spider-looking cell. From what was a round cell, it suddenly grows fingers out. And these are basically just fragments of a bone marrow megakaryocytes, and they can only circulate for about 10 days in the blood, so much shorter compared to red blood cells, which has a lifespan of 120 days. And the production of erythrocyte is regulated by thrombopoietin, and the main function of platelets is hemostasis. So how to stop bleeding. So that's basically the definition of hemostasis. Please don't get it mixed up with homeostasis, which is basically balance. When we talk about hemostasis, this means stopping bleeding. So what we can find in the body is that our platelets just go round and round the circulation until we come across a problem, we come across a wound, or we come across a disconnected area in the blood vessel. And when it comes across these problematic areas, then the platelets will release chemicals to create the blood clotting cascade. Okay, and then so now that we've talked about the components of blood, which is the cell and the plasma component, we'll now talk about the individual functions of blood, mainly transportation and hemostasis. So when we talk about transportation, there's the first thing we need to touch on is the role of proteins in transportation within the blood. So as we've talked about before, I think in the vitamins and minerals lecture, there's a lot of different minerals that we know are carried around by proteins. Now, apparently, these carrier proteins don't only carry minerals, but they can also carry a lot of different things, like, for example, hormones, and it can also carry fatty acids, or it can also bind to hemoglobin and protoporphyrin. So there's a lot of different functions of these protein carriers, with a lot of the jobs of carrying is done by albumin. As you can see, it is a protein carrier for a lot of different ligands. So that's the first function of blood, is that it carries the transport proteins, which are responsible for carrying a lot of things in the body. Now, the second transportation function in blood is oxygen transport, and this is a very crucial job, or a crucial task for the blood to perform, and it's performed specifically by the red blood cells, or erythrocytes. As we talked about before, each red blood cell is composed of hemoglobin, 250 million hemoglobin per red blood cell, and each hemoglobin can carry a heme group which contains the iron. So here we can see this is the red blood cell. This is one of the heme groups in hemoglobin, and when you look closer, this is how it looks. So each polypeptide in hemoglobin contains heme, and heme has the portion capable of binding to oxygen, because heme is a metalloporphyrin. What is metalloporphyrin? Well, first of all, let's define porphyrin. Porphyrin is basically an organic cyclic molecule. So organic means it's composed of carbon, hydrogen, nitrogen, and cyclic means it's round, as you can see, sort of round. And then it's a metalloporphyrin, which means it contains a metal right in the middle here. And the metal here, as you can see, is an iron metal. Now you can see, and then it's surrounded by the 4 pyrrole rings, which makes the whole metalloporphyrin. So this metalloporphyrin we call heme, of hemoglobin. Now, so basically when we talk about the reason why heme is because firstly we need to form the porphyrin, and then we can add the iron into the porphyrin group in order to make it heme. So we don't call it heme when it doesn't have an iron in there. Once it has the iron, then we can call it heme. And this is performed by a process called chelation. So chelation is basically the process of adding a metal into an organic molecule. And here we need the enzyme ferrochelatase to perform the chelation properties we need to do in order to turn a protoporphyrin into a heme group. Okay, so before we go into hemostasis, sorry, just a little bit of a problem here, but I would just like to continue with heme. So in order to make heme, there's a couple of different steps. I like to divide it into 4 steps for simplification purposes. So when we talk about making heme, step 1, to make a heme group, we need to form ALA. So ALA stands for alpha aminolevulinate. And then the second step is make the porphobilinogen. And then the third step is make the porphyrin. And then the last step is to add a metal into the porphyrin in order to form the heme. Now let's go through the steps one by one. So now we're going to talk about how do you make heme. Now the first step is to form ALA or alpha aminolevulinate. So to make an ALA molecule, you need succinyl coenzyme A and glycine amino acid. When you condense these two, you then get ALA or alpha aminolevulinate. And then in order to make the porphobilinogen, which is here, you will need 2 molecules of ALA. So 2 alpha aminolevulinate, and you condense these together with the enzyme aminolevulinate dehydrase in order to form porphobilinogen. And then once you have porphobilinogens, you're going to need 4 of them. You're going to need 1, 2, 3, 4 porphobilinogens to make a linear tetrapyrrole. So here we're talking about 1 linear molecule consisting of 4 porphobilinogens. Now remember that porphyrin is cyclic. We don't want a linear molecule. We need to make it a cyclic molecule, a round molecule. So there's more processes to come to make the porphyrin. We're not done yet. Now the first thing is we need to convert this linear form into uroporphyrinogen 3. Once this is done, so now it's cyclic, but it's not finished yet. We then decarboxylate. We perform decarboxylation of the acetate side chain here to form coproporphyrinogen 3. So as you can see, the A group here is gone. And then we need to then convert the coproporphyrinogen 3 into protoporphyrin. And once we've got this protoporphyrin, we can then add the heme group. So this is not a heme yet, it's only just the protoporphyrin. In order to make it a heme, remember, we need to add the metal. And in order to add the metal, we need to perform a process called chelation using the enzyme ferrochelatase to make a heme group. And now this is pretty straightforward process, but any problems associated with the enzymes that you've seen responsible to make this heme group can actually result in significant clinical problem that we know as porphyria, which is a disorder characterized by porphyrin buildup. However, we won't be talking too much about porphyria at the moment because this will be talked about in our systems block, which is basically the hematology systems block. Now we need to also talk about how heme is broken down. So we know that how heme is formed through a process of making porphyrin first and then adding the metal group. But how is heme degraded, and then how is it removed from the body? As you know, red blood cells only live for about 120 days. So what happens after that? Basically, red blood cells will be phagocytized, or they will be eaten by our phagocytes in the reticular endothelial system, and that would mean the spleen, liver, or bone marrows. So what happens in this RES system? What happens to the red blood cells? Well, basically the heme group of the red blood cells are converted into bilirubin. So that's how we get rid of heme. We have to first break the heme down into bilirubin. And in order to do that, we have the heme group here. We first need to turn this heme group into biliverdin. So you have the heme. By the enzyme heme oxygenase, we turn this into biliverdin, and then we reduce this into bilirubin by the enzyme biliverdin reductase. And once it's a bilirubin, it is then transported to the liver by albumin. As you recall, albumin can also carry bilirubin, and then albumin will carry bilirubin into the liver, where it is first converted into bilirubin glucuronide, and then once it's in the form of bilirubin deglucuronide, it can then enter the bile duct, and once it enters the bile duct, it is excreted as feces in the form of stercobilin. So it can be excreted through our intestinal or digestive system in the form of stercobilin, and this is what gives fecal matter the color. Meanwhile, it can also go into the circulation and then enter the kidneys and then get excreted as urobilin, which gives also urine its distinct yellowish color. Okay, so that's basically all that we need to talk about regarding heme and how it's formed and how it's degraded. So next we're going to talk about the function of hemostasis. So how is blood responsible for stopping the bleed? So there is a special component in blood that is responsible for stopping the process of bleeding when there is a physical rupture of the vessels. So there is basically what happens when we get a cut. For example, you're in the kitchen cooking and you're cutting onions and suddenly you cut your fingers. So there's a special process that needs to happen in order for you to stop bleeding. So the first one is called primary hemostasis, and the second is secondary hemostasis. So exactly what is primary hemostasis? So this is the first response. It's an emergency response, but it's not permanent, and it's quiet, and it's not stable enough. So it's a first response that stops the bleed, but it's not good enough. So how does this happen is first through vasoconstriction and then by the formation of a platelet plug. And we'll talk about that later. Now, in order to really stop the bleeding permanently, then you need a secondary hemostasis process, which is called blood coagulation. So here, let's talk about the processes one by one. So when we talk about primary hemostasis, what happens when you first get that cut? Firstly, your blood vessels will respond immediately by performing vasoconstriction. When there's a physical rupture in your endothelial wall of your blood vessels, then there is going to be a special chemical release, and it's called thromboxane A2, and this will initiate a process called vascular spasm. So your vessels will just contract or vasoconstrict to reduce the blood flow, so to reduce the amount of flow into the wounded area. When we reduce the amount of flow, we can transiently reduce the blood loss. And then once we've got that down, there's going to be other chemicals released by the wound, and these are ADP, collagens, or von Willebrand factor. And what happens is that these chemicals will activate platelets. So the platelet that we know used to be round and circular will turn into sort of like a spidery looking cell, and this will aggregate and stick together to form an occlusive platelet plug. So this is what we call the primary hemostatic plug. It will just clot around the area, blocking the way, making sure that the blood does not escape from the wound by plugging the area. Now, the problem is that this plug is not very strong, and when we actually have a stronger blood flow, then this can easily just break the plug. So this is why we need a process called blood coagulation to reinforce the plug and really make it stick. So we need to make a fibrin mesh over the top of the primary hemostatic plug. And this is, in order to do this, we need to activate the coagulation cascade. So the presence of a platelet plug or an activated platelet will activate the tissue factors, which will then initiate the coagulation cascade to activate the thrombin and form the fibrin mesh. So here we have the coagulation cascade itself. So it's a cascade. So a cascade means a series of enzymatic reactions. So it's step-by-step enzymatic reaction. Now, most of the factors here, so these are the factors, they're expressed in numbers. So this is factor, for example, this is factor 12, factor 11, factor 9, factor 10, factor 7, factor 10. Now, these factors, as indicated in numbers, are basically mostly produced by the liver, and it can be divided into two main pathways. Here we have the intrinsic pathway and the extrinsic pathway, and these will merge into what we call the common pathway. So intrinsic, extrinsic will merge into the common pathway. And the end goal, so what do we want to make from all these pathways, is that we just want to make the fibrin. That's what we're trying to produce, because the fibrin is like a mesh that will just stick onto the platelet plug and make it permanent and to occlude that area in a better way. So basically, the intrinsic and extrinsic pathway is usually triggered by slightly different mechanisms. So the intrinsic pathway is theorized to be triggered when blood comes in contact with a foreign surface, and then while the extrinsic pathway is triggered when blood comes in contact with damaged tissue. Either way, the activation of intrinsic or extrinsic pathway will result in the activation of the first factors in corresponding pathways. So in the intrinsic pathway, the first factor activated is factor 12. While in the extrinsic pathway, the first factor activated is factor 7. So just remember that. Intrinsic starts with 12 and extrinsic starts with 7. 7 will then activate 10 into 10A. So 7, 10. Meanwhile, here we have 12, 11, 9, 10. So it's a slightly longer process compared to the extrinsic pathway. Either way, whether you want to go through 12, 11, 9, 10, or you want to go through 7 to 10, it will all merge into the activation of factor X into factor factor 10 into factor 10A. Factor 10A will then activate prothrombin into thrombin, and then thrombin will then activate fibrinogens into fibrin. And once we have fibrin, we have the hemostatic plug. So this is just an animation on YouTube. I'll just link this video in the description because I'm not sure I can play it, but it plays a really nice animation on on how exactly does it look. Oh, actually, it's playing right now.
In a first step towards clot formation, platelets are recruited to the site of vessel injury by now exposed molecules of the vessel wall.
So as you can see, when we have sort of a hole in the endothelial cell, the platelets will be called upon there. So after vasoconstriction occurs, the platelets will come over and they will just release, after the release of several factors, and they will start to just accumulate on that area. And this is what you can see is an activation of the platelet. It starts to look like a spider sort of cell instead of a round cell. It looks like a spider, and it becomes very sticky on when it's activated like that. And once we have all these sticky platelets aggregating together, we'll then activate what is called the coagulation cascade. So as you can see, these activated platelets will release more factors that will activate the coagulation cascades later. So there's a form of communication going on between these cells. Okay. This is what we call the primary hemostatic plug, because it's just formed by the platelets sticking together and there's nothing really putting them down. And here, as you can see, they're starting to activate the cascade. Once we have thrombin, thrombin will then activate fibrinogens into fibrin. And then when we have the fibrins, this will form a mesh. So we saw the thrombin before. As you can see, these yellow fibrin strands are coming across and sticking those platelets down, and this is the secondary hemostatic plug. So once you have the fibrin mesh over that, you have the secondary fibrin plug. Okay, so that's done. So that was the hemostatic mechanism. Now let's talk a little bit about blood typing. So blood typing, there's actually a lot of ways that we can categorize people's blood types, but the most common system used is the ABO system and also the Rhesus system. Now, when we talk about when we talk about blood typing, we are actually trying to differentiate individuals based on the antigens or the protein that is present on their red blood cells. So in the ABO type, we have how does it exactly work? Is that each blood group is designated by the presence of 3 different glycoproteins, which is the A, B, or O antigen. So in your blood cells, in the red blood cells, you can either have the A antigen, you can have the B antigen, or the O antigen. So the O antigen itself is common across all types. So as you can see, this is the O antigen. The O antigen is also called the H. So just don't get confused here. This H also means O. So we have the O antigen here, which is an oligosaccharide foundation. So we all have this, but in some people, but in the A, so in A individuals, we can actually have an additional moiety here. We can have an additional group called N-acetylgalactosamine. And this is what gives the A people their A antigens. And then in B people, we have galactose right at the end here. Although the basic structure of H antigen is similar between the three groups, we have an additional group here for A and an additional galactose here for B. And the reason why the O individuals don't have that is because we actually have a mutation. So I'm an O individual. So O phenotypes occur as a result of a mutation in the O transferase, which means we don't have the enzyme, which is glycosyltransferase, that is required in order to add N-acetylgalactosamine or galactose, D-galactose, onto the H antigen. So that's the reason why O people don't have that. Now, when we talk about antigens, the reason why the ABO typing system is very important, because this is what is used mostly for blood transfusion. The reason why is because when we talk about antigens, there's usually antibodies. Now, when someone is a type A phenotype, that means their red blood cells have the type A antigens. What does this mean? It means that naturally they will also have natural antibodies in their plasma that will be directed against type B and type AB blood cells. Why? Because they have never seen a B antigen before, which means when you give them a B antigen, they will create B antibodies. They will realize that these B antigens are foreign and they will just attack those. And the same goes for B types. When we have B type blood, it means we have antigen B in our red blood cells and no antigen A, which means when there is an A antigen coming in from a transfusion, we will form A antibodies which will attack, right? And that will create problems because this will create the process of red blood cell lysis. Now with type AB, because type AB has both A and B antigens, people with this blood type will not form A antibody and will not form B antibodies, which makes them universal recipients because they can be transfused with either A or Bs. Now, meanwhile, with O, because O does not have B and it does not have A, whenever an O individual receives an A or a B type blood, then it will attack. So it will form both A and B antibodies. This means that O can be universal donors, but they are very selective about receiving and can only receive from another O blood type. Now, another blood type that we need to be aware of is the Rhesus type. So this is the second most important typing. So the Rhesus antigen is called the RHD because it's the Rhesus D antigen. So people with Rhesus positive phenotype is actually expressing the D antigen on their red blood cells, and people with Rhesus negative do not express the D antigen. So because these are not typically naturally occurring, they are usually developed after pregnancy or other sensitizing events. These are usually tested on these special circumstances, so they're not routinely tested, not as common as the ABO types. Okay, so that's all for our blood chemistry lecture. These are the references that you may wish to read up in order to improve your knowledge on the topic. Thank you very much.